Yes, yes. Okay. So I want to tell you about a percolation transition in slime molds, and it's going to be a topological phase transition, and I hope that cover will turn out to be topologically invariant. So what is it going to be about? We take a slime mold, shear it vigorously, tear it apart, and you have small pieces of the mold which are about on the order of 100 micron in diameter, and they then fuse, they start walking, they fuse together and build an extended network. The scale is here 10 centimeters. And that takes about 24 hours. And what interests me is the point where you go from small disconnected pieces to a large component spanning the whole size of the network, and that's what you call percolation. You also notice that there's a hierarchy in these veins. There are main veins and smaller ones and so on. That's a feature I'm not going to talk about today, but obviously is something which is shared by many transport networks. If you look at the vein network in leaves, or the drainage network in a river system, or in fact the vascular system of our bloodstream, and I come back to the vascular system later. Networks also are ubiquitous, and what we are interested in is their nodes. So we are not interested in any geometry, so I'm really interested in how they are connected. And the simplest, and one example is, for instance, a railway system where you transport not matter, but living matter, people. And here you can see very clearly what's going to be the points we are studying is the kinds of nodes. You have end nodes where the line ends. You have nodes with two links, like here. You have nodes with 3 links and one with 4 links. And that's as far as we go. Of course, there are higher hubs. It turns out the supposedly biggest hub here is in fact a bunch of dead ends in this case. As I said, networks are ubiquitous. You would have them in chemical signaling pathways in the cell, or in metabolic networks, or how the chemicals are broken down. And in each time that there are sort of relatively simple nodes with small link degrees. What is a particularly interesting and funny and nice example are social networks. Here you have the sexual network in Jefferson High School in the States. And the most people, 63, just have a binary relationship. But there are others. There are sort of active girls, very active boys, like this. And the reason I'm showing this is because here you see a giant connected component, like you had at the end in the Physarum Polycephalum. And that's where I want to go. To explain you how you go there, and why it's important here. Well, if you have a giant connected network, in terms of contagious diseases, you don't get AIDS here, you get AIDS here. So let's go back to slime molds. Slime molds form networks to search for food, and so they are very large. In fact, they are the largest cell on Earth. What you see here is a petri dish, and there's one cell in it. The cell has a multitude of nuclei and spread organelles, but just one continuous plasma membrane. So they are sort of growth fronts and tube-like structures. These tubes show shuttle streaming. Okay, so what you see is a sort of a oscillating, very periodic contraction of those tubes, pressing the cytosol through those tubes, and what you can see, what you see flowing by are nuclei, mostly in mitochondria. And then there's a mantle of actomyosin around these tubes, which drives that. And as I say, the typical time scale of the system is 2 minutes, 1 to 2 minutes. This is the time scale of the oscillations you see here. So it is, as I said, it's a single cell, as opposed to dictyostelium. If they would go to macroscopic sizes, that there would be accumulation of several ones. So this is called the acellular. Pardon me? This is sort of one of those or less, in fact. Yeah. Can be on all. You see, they can come in all sizes. So to tell you where it's located in the evolutionary tree, it's not the same than a fungi. It is a separate kingdom from animals, fungi, and plants, but it has a homology, very strong, very large homology in terms of actin and myosin. They're not the same, of course, but there's an actin-myosin system there. The life cycle, they mostly live in a plasmodial state where they have, in the form I just showed you, they live happily there, have thousands or millions of diploid nuclei, and then for reproduction they build spores, which then are uninucleate and are sort of more well-known amoebae. Then they have sex, and they grow again. They grow by division of the nuclei, but the cell isn't dividing. So just the nuclei are dividing, and so it grows and grows and grows, and proportional to its size, it gets more nuclei. We're going to use this site here because we're going to disrupt the cell, basically from going from here, throw it back, and use its propensity, to grow and infuse together to come to here to see what's going on. So that's what is here. So you had this slime mold, you cover your petri dish with water, then they float off. You take it in a shaker, you shake it, and you plate it. You plate it on an agar. You wait till excess water has evaporated. You plate it in a water pressure controlled atmosphere, and then you wait and you look at it. And in fact, this is done with a Canon camera. Okay, so upper scale Canon camera, but not these are large objects. We are only interested in the skeleton, so just in the topological ways these networks are connected. And so what you see here is image analysis. You see in each of these things, you see a black line. If it's sort of one node only, it's just a dot. If it's sort of elongated more than its width, then you have sort of a line with two ends and more complicated connections here. And we always denote the largest connected component red. Okay? So what you see now is going to be a movie of that dynamics, where the largest component you see in red. Okay, so it takes a while. It gets larger and larger, and somewhere between around 7 hours, it suddenly spreads to the size of the whole network, and that's what percolation is. Okay. Yes. We haven't looked at loops, but something interesting we are looking at that right now. Okay, so that's how it is. And now, how do we do the analysis? As I said, we are only interested so far in those nodes. We take singly connected nodes and call the probability to find such a node P0. The endpoints we call P1. Then the physiological P2 makes sort of no sense because I don't quite know where a station with two connection points should be. So they are absent. We set them to 0. Then there are the nodes with 3 connections. and occasionally there's a 4 node. And here we call it a 4 node if if the 3 vertices are closer together than the diameter of the tube. This is a little bit fishy, but I think that's a working hypothesis. Okay, now what you're going to see is the dynamics of these probability functions distributions over time. Red and green is P0 and P1, so they go down. There's fewer and fewer not connected pieces. The ends grow. The P1s go down as well at the extent of the triple connections, right? So this is what in the end, the most of the network is actually consisting out of P3 nodes and a few P4. Now it turns out that the percolation, and I will work it out exactly, happens at the ratio of P0 to P3, which is plotted as the black line on the right axis at 2. At exactly 2, the percolation happens, and it's around that percolation point where the P4 go up. So as you see, the P4 are much smaller than the other ones. So in the first approximation, we set them to 0. So we say, okay, all our network has maximally P3 connections, and that's what we work on further. Now, what do we want to find is the fraction of the giant component, which is defined as the size of the largest component counted by the number of nodes, over the size of the whole network, it has the number of total nodes in here. If you have a finite giant component which does not grow and does not scale with system size, then in the thermodynamic limit this goes to 0. Whereas if it spans the network, and if you make the network larger, it scales with the network, then you have a finite fraction of the giant component, and then this transition from 0 to some finite number is called a percolation. You can already see that in a finite system, you're always going to have somewhat from 0, a little bit of S we have already because we cannot get rid of the finite size effects, right? And so that's what you see here. As you go along, the S is 0 here, and around here it will go up, and you have percolation. Okay, here's the data of 50 of those movies I showed you. These are the blue curves, and this is the median. And around P0 over P3, it goes up. So this is the value of 2. Now, can we understand this 2? There's you visit the theory of graphs, and it's all done for you. Well, I was amazed that I haven't found the solution we presented in the PhysRef letter. I just found the axioms to do it. Obviously, I think the reason it wasn't there is people are interested in more complex network, large K, more numbers, and not in the small link degrees. So we are interested in small link degrees. And so what you can use here is a self-consistent relation, which is actually valid generally. So S was the number, the fraction of the giant component. So 1 minus S is all the rest. Sort of U is the probability not belong to the giant component. Now there is, Newman has given in his nice review self-consistent relation, meaning not to belong to the giant component is equivalent that all your neighbors all your connected neighbors do not belong to the giant component as well. This gives you a self-consistent relation like this, and all your neighbors, you just sum up all those possibilities you have.
Oh, they forgot to me there.
No, they're there.
In this equation as well?
I think so, yes. I think it's true. So now you take the self-consistent relation, the normalization condition, assume that all pk vanish for k larger than 3, and solve it for that ratio p0 over p3q. What you get is a cubic equation which you can solve exactly, And bingo, you get, here's the analytic solution, but what we're interested in that it's non-zero for values smaller than 2, and it goes up continuously in sort of a second order phase transition. And as I say, and it's pretty convincing, at least I think so, that they follow this line. Of course, there are some the S is different from 0 in this regime, and I told you that is sort of finite size effect. So here's a trajectory of the movie you saw in the beginning, just the skeleton. Here's the S as a function of this Q parameter. Now now it shoots up when the giant component goes to the size of the whole network, and you can see, of course, the system has some fluctuations. Now, it doesn't stop here. We found a paper by Molloy and Reed where they give a topologically constrained at percolation, which is this peculiar weighted sum, which in our case means that there are 3 times more P1 links than there are P3 links. And if you think about it, if you have more P3s than that, then you percolate. If you stare at it at some time, you think at some point you understand it. Now, with this additional condition, which is valid just at percolation, you can in fact calculate the probability distribution as a function on k, which just consists of 3 numbers. They come out like that, and you can calculate the average link degree at percolation. At percolation, it's 1. And so this linear term vanishes at percolation, and it deviates with a slope of minus 1/6. So let's see how that compares to experiment. P1 and P3 look quite good. There's always some dispersion here. P1 is a little bit more widely dispersed, but also sort of more on the right and not on the left at percolation. And as I said, P4 is found to be not equal to 0, but compared even to P2, it's almost an order of magnitude smaller. So it was in the first approximation okay to forget about it. Now, if you look at the mean link degree, which is this is again our 50 contours, and indeed all those 50 slimy molds obey topology and go through the point 1, 2 with a slope of minus 1/6. So this is a result, and I guess one could get the quadratic term as well, but we didn't do so far. Now you scratch your head and say, oh well, I can also solve a quadratic equation. So let's lift that P4 to P0, and let's just say everything larger than P5 or in equal P5 is 0. And then in fact you don't get a transition point, you get a transition line. You have two parameters instead of P0 over P3. You now have P3 over P4 and P0 over P4, which we call P and Q, and you get this line which shows you. So on the right you have non-percolated systems. On the left you have percolated systems, and the color coding tells you how big the fraction is. And you know that, of course, you have particular trajectories, and we haven't checked whether they cross here a specific point or not. They simply they cross it at some point, as we already knew from in the first place. Now, I want to tell you that this has a lot to do with cancer. Here's a review about the hallmarks of cancer, and I don't want to go through all this thing, which is not of interest here, but one of it is inducing angiogenesis. So the tumors, they need blood to have oxygen and nutrition, and they derive that from the existing circulation system, and for that the microenvironment of the tumor is very important. So let's briefly look into that, what's going on here. so what has been known for decades is that, if there's a tumor somewhere and there's a nearby blood vessel, that there are growth factors, and then the blood supply connects to the tumor in a process called angiogenesis. So, but that's just one mechanism of vessel formation. So angiogenesis is the extension of pre-existing networks by sprouting, as I just showed you. But there's also vasculogenesis, which is de novo formation of vessels from disconnected blood islands like in embryos. So that has been found about 10 years ago, that in fact there are a line of aggressive tumors which do not get by sprouting, but they make their own network. And this is similar to, that's why they call it vasculogenic mimicry, to the growth of the vasculogenesis in embryos. So if you look at an egg yolk in the first few days, you see sort of blood islands appear, which are disconnected. And within already day 3 or so, they are connected already in a complicated network. And of course, they have to percolate at some point here. And that's sort of the analogy we draw. And so what's happening here, also from that review, well, you layer on your egg yolk, and then you form these blood islands, which consists of precursor blood cells and walls of the arteria, and then they fuse, and finally build the vascular system. And so, in some work by Folberg and people later, in a tumor of the eye, they do find that there are channels in the tumor generated actually by tumor stem cells, which has been shown 5 years later by another group, and they connect to these choroidal vessels which have been there before, and so this is sort of the cartoon. Instead of having sort of going the pre-existing blood vessel growing into the tumor makes its own system via this percolation method. And the reason it hasn't been found for so long is that there were different cells. People always stained for arterial wall cells, and so they didn't see that. So that's why it has really only recently, some 10 years ago, has been found. There are some pictures here. This is one of those vessels newly formed, and here you have a pre-existing one, and you can see that there's sort of channels here. And I'll show you a increased version of that. And so, I take the face value what it says here in this paper. So these are the newly formed tumor-derived channels which connect to the pre-existing one, and they haven't sprouted out. This is not only restricted to these tumors in the eye. You also find it in melanomas, in skin melanoma cancer. Now, so we have we have found we had done this experiment with this Physarum. They were disconnected pieces, and they were percolating, and we are finding this very general description in terms of these probabilities with these very general topological constraints. So we thought, well, it should actually apply to this case as well. And indeed, we found an experiment a few years later by Serini et al., where they did in vitro experiment. So they had endothelial cells on matricelle and let them grow. And we pulled the movie out of the web. And indeed, after 4 hours, the last picture, they form sort of a two-dimensional network, which starts from disconnected cells at the beginning, and at the end you have a sort of a percolated network. And in fact, if you do this, do the analysis I showed you, then you connect into a percolated network. And of course, now we can put this into our graph. So the blue one was the slime mold. The crosses is the data of the movie I just showed you. And indeed, they nicely follow that phase transition. And it also applies to the mean link degree. If I sort of blow up that region, then indeed, a little bit more scatter here, because the movie was of bad quality, I think, at least the one which we pulled on the web. So indeed, it goes to this 0.12 with a slope of -1/6. Now, what we propose is that we might use that universality of percolation as a gauge for tumor vascularization. So percolation is independent of the detailed mechanism in 2D versus 3D growth. Now, hold your breath. That applies in the space I have described it. I have described it on the level of these probabilities, and in fact, on that level, it does not depend on the dimension. I cheated insofar. I didn't tell you any dynamics or mechanism what might determine the value of this probability function. If you come up with a theory of that, then in fact it will depend on dynamics and on the detailed mechanism. You have different universality classes. But practically, when you look at these probability functions, that it doesn't. So we can use this strong topological constraint to calibrate vascular network promotion under various conditions. So our proposal is, if you compare different therapeutic approaches which hinder the development of these blood vessels, you can actually gauge them with respect to this topological point, which is independent on the detailed mechanism. So you can compare your effectiveness of your therapy quantitatively with that. Now, let's go back to the slime mold, and let's go back to the small components we started off with, which we call microplasmodia. Okay? So remember, so this is the movie I showed you, and I want to focus now on these disconnected pieces on the left. Well, oops. Okay, so if you look at them closely, in fact, they flicker. In fact, they oscillate. At some point, they polarize and start to move. In fact, look at this one. This stays, it wobbles around a little bit. And this is going to be catched up. This one is still lonely, but now it's somehow, I think there should be some chemical signal, and it actually grows into the big parent slime. Okay, so how do these guys look? Okay, so now this is under a microscope. They show oscillations, active oscillations. This is a contraction of that actomyosin system, and you see that there's two periods. There's a quick oscillation, which is on the order of 2 minutes, and there's a slow beating on the order of 20 minutes, which you see very nicely if you look at the area. So here's the slow beating, very regular over 2 hours, with a period of pretty much 20 minutes. And then there's the big wiggle up here, which is on the order of 2 minutes. Now you can do image analysis. You get the contour, and you find the normal velocities. And of course, then these big wiggles show up as plus and minus velocities of the surface, of the equatorial surface of your microplasmodium. And the velocities we find here are on the order of a micron per second. It also grows, but it grows on a much, as you've seen, when you compare that with the other movie. This is 1 or 2 hours. It grows on a scale of a day by, let's say, 10 or 20%. So this is the projected. This is the equatorial area. Yes, this is the equatorial area. Okay, so as you can see, there are some oscillations of the frequency of this quick component at the extended state, they are actually faster than and larger in amplitude than actually at the contracted state. You can analyze this by taking the correlation functions around the maximum and the minimum, and you get a sort of these are the full correlation spaces, and these are the cases for just the time correlation function, which is got extended in this case. Okay. Now, to get the network, they should fuse. This is now speed up by factor 5 with respect to this. You still see this quick wiggle. They get in contraposition. They obviously somehow see them already, and then they take some while, and they actually fuse. Great. I was getting. Believe me, I would have just ignored you. I learned that from my former boss. That works very well. Okay. So here's the fusion. Now, once they're fused, they actually already form internal channels, which you see here, which is a beautiful false color picture. And what you basically see is a derivative of the images before. And so everything which moves very fast is bright. And so you see these are sort of two of those compartments having joined, with a period of 2 minutes, they pump cytosol in between the compartments. So you can already see here that there's obviously an internal structure. So we see this here by thymidine section of microplasmodia. So ignore those artifacts. These are sort of shear induced from the cutting. You see that they are sort of internal channels and more external ones. If you do this with the EM, these are pictures from Singapore. We have already done this now ourselves in Bremen. You can see that there's these are the ones on the outside, and they're actually connected to the external. So you see that there must be actually two different systems, internal ones and external ones. And in fact, what you see here, the remnants here is the slime. So these are the ducts of the slime. So what the system is, it's contracting, and it's a porous material which is contracting, which then they should come with some time scales with it, different time scales. So there's going to be a viscoelastic time scale from the cytoskeleton itself, and there's going to be some time scale of squeezing out that water out of that pores, which you can beautifully see here, the opening on a scanning electron micrograph here, and the sizes fit. When they develop further, they make these networks, and if you do cuts here, in fact, this is a cut through one of those veins, and you have sort of these porous actomyosin mantle on the outside, and the inside you have the tube where the cytosol is flowing, and at the rim. So these are orthogonal cuts to the. These cuts are orthogonal to the surface, right? So it's a cut along this. So this is sort of not the tube here is actually a little foot. They grow little feet into the agar, and the space where material flows is sort of extended. Now you wanna find out about their viscoelastic properties. You do this by micromanipulation. So this is done in collaboration with Ang Wei Tech at NTU Singapore. So they have a holding pipette, and in fact, when you hold these guys, they still oscillate. So which tells you have to be faster than the 2 minutes to avoid very complicated analysis. So we do this with a capacitive force sensor, which you can do with different frequencies. So we haven't explored a frequency range a lot, mainly working in the 1 Hertz regime. So if you do this, you get curves like that, sort of get the voltage out of your force sensor as a function of time, and you go up and increase the poking of the tube into the microplasmodial 10 micrometer wise. Here you go from something like 5 micrometer to 80 micrometer, and what you already see, there's a nonlinearity here. So somehow the material gets stiffer the more you punch it in. And there's on the order of 10-15%. There is a sort of a plastic compliance here, or a viscoelastic one of long relaxation times. So to get numbers out, here you can see the repeatability. So here this is one slime mold, and we repeated it with 1 Hertz 3 times. So in the low regime, it's nicely repeatable. What I plotted here is the maximum of the sinusoidal punches. If you change frequency on the order of 10-20%, you get the same slope. And what we find, if you calibrate that, you find a slope, an elastic Hooke's spring constant of half a Newton per micrometer. Now, if you do a half space approximation, assuming this thing sort of half space and a circle instead of a square, which it is, you get a sort of a nominal value for the elastic modular, which is 4 kilopascal. Now, if I plot the scaled force versus the scaled extension, so I plot. So what I show you is data for 20, 40, 60, and 80 deep punches. The black one is the one with small indentation. The blue one is the one with big indentation. And I scaled the extension, this 20, 40, 60, 80 all to 1, and the corresponding force to 1 as well. So you can see that for small indentations, you're sort of linear. And of course, you have a viscous component. And we fitted that, since we were not a lot frequency dependent. We were starting with a very simple Kelvin model. And what you get out is of course, the 4 kilopascals we found before, and a nominal viscosity of 24 Pascal seconds. What you see is the more you punch, the more nonlinear effects is going. No, you might call this strain hardening, but we haven't published this yet, because you need to worry when you. So this thing is 100 micron, and if you punch a 80 micron, you squash it about half its size. And so there's going to be geometric and surface tension effects. And we currently do a finite element analysis to check what part of that might be strain hardening and what might be nonlinear effects. Whether there was something left over. So finally, I want to thank my team. So this is Christina Oetmar. I did basically most of the experiments on the Physarum system. Adrian did the analysis, which was this percolation analysis, and Eric Burnett is the senior PhD student in the background, which supplied a lot of help to those guys. And this was my collaborator, Niels Gauthier at the MBI, who had the idea, why don't you make this interesting and talk about vasculogenesis? And I think that's what we did, and I hope I did interest you a little bit. Thank you very much.
What happens to the oscillations as the fragments fuse?
They, in fact, they synchronize, they asynchronize their phase and then fuse.
So they first somehow synchronize and then.
I think we haven't looked at this in detail. They first synchronize, and in fact, in between, we could look at the movie. They don't oscillate because there's this plate in between.
Yeah. You use the Newman theory is based on the Galton-Watson process, which is for.
Sorry, didn't get
the
it.
Newman theory.
Yes.
That you mentioned at the beginning, that you use for your, in fact, the planar network that you have is based on the Galton-Watson process that is for tree-like graph, which is supposed not to have closed loops.
Okay.
And you have a lot of closed loops in the experiment, which make me think that you have a lot of correlations, which should use something different that the Newman theory.
Since I'm not a theoretician, I wasn't so much concerned about that. It worked pretty well. No, but I mean, I hear you what you say, and
percolation, or I mean.
No, but I mean, that's a good point. But of course, what is also seen is, if it would be true exactly, if it would be true exactly, we shouldn't see distributions in the p-zeros. And then some of that distribution is due to errors in the image analysis, but not the rest of it. So maybe these are the loop corrections. But the loops, in fact, there are a lot of loops, but they are irrelevant for the percolation because, well, maybe I shouldn't say that. There are more at least there are more loops at the end of the process than at percolation.
But you can have also some kind of percolation if you think in diffusive limited aggregation, for example. So you have used graph theory, which I think I'm not that sure which is the best kind of tool that you use to analyze.
Well, I show you the graph again. I mean, these are 50 slime molds with a little bit of varying biological conditions, and they all do -1/6. How better do you wanna plot
but with
a biological experiment? I mean, let's discuss. I mean, I'm happy to use other theories. I'm not jumping on that. I was just happy to get that graph out.
You mentioned at the beginning that there are many nuclei.
Yeah.
I was wondering, when you showed these various micrographs, would we actually see several nuclei?
Yes.
The two fusion things, were they also already multinuclei?
When they fuse, they are already multinuclei. So you double the nuclei, but they are already thousands before.
Would there be a characteristic doubling rate for the nuclei?
About 4 hours they divide. But it isn't very visible in the macroscopic slime mold when they divide. They actually do it at the same time. We should look at that. But as you see in the movie, there isn't something very obvious for that.
You had these strong fluxes that drove this oscillation. So what is the role? I mean, the biological role of these fluxes. I mean,
they need. See, this is a huge organism. It can go centimeter wide, and it might find a dead body here. But all the rest of the slime mold is on the right. So you want to get the material from the left to the right. So it's communication of chemical signals and nutrition in these. They can grow actually to micrometer sizes. Actually, when one of the fathers of this field was emeritated, retired in. They grew him a dig. His wolfram. They grew him a W on several square meters on the floor. So you can get these guys really large, and that's why they need this communication.
You mentioned these experiments of endothelial cells in agarose gel or
matrix gel,
and forming a network. And you mentioned you want to transfer the what you learned from this network formation to tumors. I didn't quite get how you want to do that.
Well, the idea is that the percolations, what we used, is pure mathematics and topology, and I had 3 equations, and there was nothing going into them. So the percolation point in a tumor is going to be exactly the same than in the 2D system of the matrix as it is in our system.
This is a claim, or is it some?
Well, it is for the tumor. It is a claim, but it is in so far no claim because I showed you. Well, it is a claim, but it has to be like that because mathematics doesn't let you out.
No, I have to take into account.
I have to take into account the far right remarks at the back.
I don't think so, because I think that the network in the tumor is always percolated because it's growing already in the pre-existing network, the vascular network of the healthy tissue. The picture you have shown, which you said it is known for years, it's wrong because the tumor is not growing a kilometer or, I mean, in this case, distance from a vessel, but it's growing in already vascular tissue, and it grows new vessels via angiogenesis, and by growing it removes also vessels.
So you're telling me those 2 papers are wrong?
Yes.
Okay, we should discuss that. I'm happy to learn that. we got a note in science on that anyway.
No further question? Okay, thank you very much.